Okay, so this is Mendelian genetics. So within this objective or within this chapter, we want to be able to answer how are characteristics of one generation passed to the next, and how can individuals of the same species and even siblings have different characteristics. So first we're going to start out talking about Gregor Mendel. Gregor Mendel was actually a monk who studied over around 300,000 peas in his lifetime. So what he did in his research was discovered that genes are passed to offspring. We call this the term inheritance or heredity. This is now who we call the father of modern genetics. So how did he do this? What he did was he actually took pea plants and he cross-pollinated them. So he pretty much just played with the actual pea plant that you can actually see here. And he took the stamen, which is the part of the male, kind of the pollen, if you will, and then the carpel, which would be similar to like an ovary of the female. And what he did was he cross-pollinated it. So he took pollen from here and then put it on the stamen to actually produce offspring. So this was the offspring that he actually produced. But what you notice here was that he had a variation of different types of pea plants. So in his studies, he found that not all pea plants were produced to be maybe smooth and green. He had some that had dented seeds and then some that actually had yellow seeds. So through his research and through his studies, he was actually able to come up with what's over here on the right-hand side. Actually, Punnett was the person who came up with the Punnett square, but this is what Mendel's work allowed for. So what he did was he took a parent generation, which was homozygous dominant, which I'll get to in a minute, and homozygous recessive, and he crossed them. And what he came to find was that his offspring were heterozygous, so when he crossed it with another offspring, he would get a probability of figuring out whether or not the seeds would be all green and smooth or green and dented. So this is kind of an example of what's to come as we go through this next section. So what we have here, again, is starting off with a parent generation. So as you're going to see throughout this unit of study, we're going to cross-pollinate or mate specific types of species depending upon their allele or their characteristic. So what we have here is a tall plant with a short plant. And in order to represent that, we're using these letters here. Now, capital T is going to be dominant. So we refer to these as gametes. So going back to meiosis, when we talked about that, gametes were what was produced when you had the sperm or egg, depending upon what species type it was. So what we do here is we take the gamete from both parents, and then we figure out what their gamete would be. So in this case, their gamete would be capital T, lowercase t. And then if we were to mate two heterozygous plants, we would get capital T, lowercase t, which is heterozygous, and then we would take their gametes and then do another cross. So this again is just showing you what Mendel did within his work, and there's going to be examples later throughout this presentation to allow you to understand this a little bit more. So what are alleles? So what were those letters that were being mentioned in the previous slides? So as you guys remember, when we studied sex cells in meiosis, we found out that we have what's produced are gametes. So gametes contain their own genetic information, and if a offspring is produced, like a baby, then you're going to take those alleles, those gametes, and you are going to give them to the offspring. Now, it depends upon which gene or which allele or gamete is dominant over the other, and that will determine the characteristics of an offspring. So it just depends upon who you're or what trait you're determining for. So within a gene, within a chromosome or chromatid of an organism, each chromatid can have a specific trait. So in this case, this is where height would be. And this is where the P gene locus would be. So a locus is just a spot on an allele where we would find that specific trait. So you heard me say terms prior in the previous slides. Homozygous dominant, heterozygous or hybrid, or homozygous recessive. So when I say what are the alleles for an organism or gametes, you have options. Homo, meaning same, zygous, meaning your embryo, your offspring, and then dominant means capital. So homozygous dominant, these two capital letters are the same. Zygote is referring to the offspring, and dominant meaning their capital letters. Hetero means opposite, zygous again meaning the offspring, or hybrid. Hybrid means like a blending. So here I'm going to have one capital letter, one lowercase letter. And notice within this state, you always want to write the capital letter first. Then we have homozygous recessive. Homo meaning same, zygous meaning offspring, and recessive meaning it is lowercase. So here, that's what we have here. So notice the notes at the bottom. Dominant alleles are always expressed unless under special circumstances, and we'll learn those special circumstances later within the unit. And we have recessive alleles are always masked unless they're in the homozygous recessive state. So what this is telling you is if my offspring is homozygous dominant, or if it's heterozygous, I'm always going to show whatever the dominant trait is. And then if I'm in a homozygous recessive state, I'm always going to show whatever that lowercase recessive trait is. And you'll see examples of that in the slides to come. So what happens now is we're actually going to solve the Punnett square. So if we have a problem such as this, actually let me go back. So when you're solving problems, in this case, we're going to be solving a monohybrid cross. What you're going to do is you're going to place the dad's alleles here. One letter is going to go here, one letter is going to go here, and then the mom's allele, one letter will go here, and then the other one will go here, and then you'll put your offspring here. So as we go through this, you'll see what I mean. So here's our problem. If you have a homozygous dominant father, so homozygous dominant, homo meaning same, dominant meaning capital, who has brown eyes, mates with a heterozygous female. Hetero means different. What is the probability that the offspring will have brown eyes? So if we're talking about the color brown, we'll probably most likely use the letter B, but in this example, I'm actually going to use the letter A. But again, let's go back to the father's alleles, because remember, the father's alleles are going to go here. So he's homozygous dominant. Homo meaning same, zygous, and then dominant, so we're going to have a capital letter and a capital letter for the dad. The female is heterozygous, so she's going to be one capital letter, one lowercase letter. Now, how we complete a Punnett square is you're actually going to bring the two letters into this box. So I'm going to bring this letter here, and I will get something that looks like this. So two capital letters. And then for this one over here, I'm going to bring this letter down, and then I'm going to bring this red A all the way over here, so I would get something that looks like that. Now to fill in this box right here, I'm going to take the capital A and bring it all the way down here, and I'm going to bring in this lowercase a. Now remember the rules, the capital letter always goes first, so that's why you see it depicted here. So what I'm going to do right here is again bring this A all the way down, and then bring this lowercase a all the way in, and I have this offspring. So again, capital letter always goes first. So what does that mean? Now, if I want to know the probability of the offspring having brown eyes, I have to take a look at what's inside the boxes. Now, two capital letters means it's homozygous dominant. So again, this would be homozygous dominant. And down here, I have heterozygous and heterozygous because one is capital, one is lowercase. Remember the rule that if as long as it has one capital letter, so this has a capital letter, this has a capital letter, as well as this one and this one, my eyes are all going to be brown. So if I were to write this in determining like percentage and ratios, I would answer the following questions. How many have brown eyes? I would say 100%. Now the ratio for that, so probability usually is in reference to a percentage, but the ratio is about how many per total. So what I would have here is 4 out of 4. So 4, meaning all of them, are going to be that specific trait, in this case brown eyes, out of 4. So 4 out of 4. Okay. So moving on, there's some other questions that can also be asked in reference to what we call genotype and phenotype. Genotype is referring to the letter combinations. Phenotype refers to what I see. So the genotype, again, is what is found within the genes of an organism, and phenotype is what do we see on the outside. So genotype, I would determine the percent and the ratio for each circumstance. So how many are homozygous dominant? I would have 50% or 2 out of 4. Heterozygous, I would have 50% or 2 out of 4. And homozygous recessive, I would have 0% or 0 out of 4. So those are my genotype percentage and ratios. Now for phenotype, I would have 100% be brown-eyed or 4 out of 4, and blue eyes. In this case, I would have 0 or 0 out of 4. So again, this is a very simple monohybrid cross. Monohybrid means we're dealing with just one specific trait. Now moving on, we're going to bring in what we call a dihybrid cross. Di meaning two. So this is where we're not only just taking one trait into consideration, we're actually taking in two. So what we're going to do here is the problems are going to be a little bit longer. We have to follow a couple of different things in steps in order to find out the answer, but the same result will take place is we will end with percentages or ratios depending upon what the question is asking us. So let's go ahead and look at the question. It says, suppose that black hair, capital B, is dominant over blonde, lowercase b, and brown eyes are, capital E, are dominant over blue eyes, lowercase e. So now here we set up who our father and our mother are. So it says the father has black hair, and he's heterozygous for the trait, so it actually specifies what we are, and brown eyes. So he's black hair, but he's heterozygous, so that would mean he's a capital and a lowercase, and then he's brown eyes, and it says he's heterozygous for that as well, so he would be capital E, lowercase e. And now we move on to the mother. The mother is blonde and blue eyes. So if she's blonde, she contains the recessive alleles. And in order to show those recessive alleles, she has to have two of the same letters. So she's both lowercase b. And then again, blue eyes is the recessive trait. So she has to have two lowercase e's. Now, these are the alleles that we will use, but we actually have to turn them into what we call gametes. Now, gametes are the combination of these two alleles mixed together. So what we do in order to figure this out is a method called FOIL, which is the first, the outer, the inner, and the last. So what we're going to do for the dad is we're going to perform FOIL, and we're going to figure out what the gametes would be. So what we do here is first. So I'm going to take this B and mix it with this E, and I will have my very first gamete combination. Then I will take the B and go to the outer E. So that's outer. Then I would go inner, so this B to this E, and then last, this lowercase b with this lowercase e. And then I would do the same with the mom, with the female. So first, outer, inner, last. So let's refresh. Alleles are just what does the word problem say? So the dad, he was heterozygous for black hair and he was heterozygous for brown eyes. Now, when we did the FOIL method, what were his gametes? So his first gamete would be capital B, lowercase e. Oops, sorry. Actually, let's do the mom's alleles, and then we'll go and list the dad's gametes. So he would be capital B, capital E, capital B, lowercase e. Actually, I totally did those arrows wrong. Next would be inner, so that would be here, and then last, I completely butchered those arrows. I will fix them on the presentation, but hopefully you guys know first, outer, inner, last. I completely did not do those arrows right. I apologize for that, but hopefully you get the gist. And then the same thing over here, because they're all the same alleles, you're going to end up with all the same gametes. So moving on, what we're going to do now is we are going to take into consideration all of the information that we had on the previous slide. So what we're going to do now is notice we're going to set up a dihybrid cross. Now, because it's taking in an extra allele, that creates a larger Punnett square for us to figure out what the combinations of offspring will be. So instead of having 4 potential offspring, we would have a potential of 16 different scenarios that can take place. So what we're going to do here is the same concept. We're going to take what's listed here and what's listed to the left and bring them together. Now, the rules for this is that you have to keep like letters together, and you have to make sure that the capital letter is always listed first. So for this first box here, we would have capital B, lowercase b, capital E, lowercase e. So notice that there's a total of 4 letters. Capital letter always goes first for the B's, as well as for the E's. Now notice, if I take the capital B and E and I mix it again with a lowercase b and e, it's the exact same thing that was listed above. And that would mean that the last 3 boxes are going to be the same as the first, so those would all be the same. Now if I go to solve the next one, I have a capital B, I have a lowercase b, I have 2 lowercase e's, so it would be capital B, lowercase e, lowercase e. And again, remember, they're all the same in this situation, so those would all be the same. Moving on, I have 2 lowercase b's, a capital E, and a lowercase e, so I would write it like that. And again, fill in the blanks for the rest of them. And then down here, I would have all lowercase letters, and that would be true all the way down. So if you feel like you have to work them out, please do so, but make sure that you are able to understand what's going on here. And if you don't, be sure to ask. My whole Punnett square, my dihybrid Punnett square or dihybrid cross is complete. I have a series of questions I need to ask. Or answer. What percent of offspring will be totally heterozygous? The word totally means out of all 16 of your offspring, which ones will be heterozygous? So heterozygous, remember, is 1 capital letter, 1 lowercase letter. So I have 1 capital, 1 lowercase, 1 capital, 1 lowercase. So that would be 1, 2, 3, 4. Okay. Any others? I don't see any. So 1, 2, 3, 4 out of a total of 16 would be what percent? 25%. Now it's asking for the phenotypic ratio. So phenotype, remember, is what we see outwardly. So what are the possible combinations that we can have? We could have black hair, brown eyes, black hair, blue eyes, blonde hair, brown eyes, or blonde hair, blue eyes. So looking at this first column here, this would be black hair, brown eyes, right? So I have 4 that are black hair, brown eyes. In the next column, next 4 right here, I would have black hair, blue eyes. I would have blonde hair, brown eyes. Then I would have blonde hair, blue eyes. So I would have 4 out of 16 for each of those scenarios, or 25% for each. But again, it asks us for the ratio. And then it says, what percent of the offspring will have blonde hair and blue eyes? So blonde hair would mean I have to have 2 recessive letters, and then blue eyes would mean I'd have to have 2 recessive letters. So I'd have to have 2 lowercase b's, 2 lowercase e's. So I have here 4 out of the total, which would be 4 to 16. So I would have 25%. So this hopefully is just a reiteration of what you've learned through the Amoeba Sisters, but I just wanted to make sure that you had instruction for me explaining it to you, and that way you could actually consult it and come back if need be. So this is just something in addition to help you understand this concept. So I hope it's helpful.